Thank you. So I'm really pleased to be here. I'm actually a committee member of the Computer Arts Society, so I've been working with them for a while. And yeah, I got a bit of a background to show you some of my previous works, just a rough idea of like all the different directions. And then I'm going to talk about a project called The Mutability of Memories and Fates, which I did with the Helmholtz Centrum in Munich, and then how that led on to the collaboration here. Because the way my work goes, it's always building on the previous and in different directions. So yeah, I've actually made quite a lot of works with robots as well. So here's some of the humanoid robots that I've created. And I think back in 2004, I became the artist in residence at the Centre for Computational Neuroscience and Robotics at Sussex University, where I took over from Paul Brown, that many of you will know, who used to be the chairman of the Computer Arts Society, as their artist in residence. And so we've got Harry, humanoid art research robot 1, who's a robot that gets bored of you, and so you must entertain it. So it's like a comment on basically how we use technology. But we've done lots of different events with that. And then also Cyberspecies Proximity Robot, which is about how we share our cities and sidewalks with embodied humanoid robots, or how we might do that in the future. And that's just there at ZKM in Germany as well. So it's been shown in lots of different locations. But as Bronac mentioned, I love bacteria. Can't tell you how much I love bacteria. And this is my plague dress here, which is a 1665 dress design and impregnated with the DNA of Yersinia pestis, which is bacterium that causes plague, which I extracted myself in the lab at Colindale in London at the UK Health Security Agency. And I've also worked at Porton Down, where they also have other samples and things. And one of the bacteria from my body, actually from my nose, is in the collection, National Collection of Type Cultures. So you can actually get it and use it in your artworks in the future, maybe. You do need a Category 2 lab. I'm not the first person to put a bacterium in the collection, though, from their nose. They've got Micrococcus luteus from Alexander Fleming's nose. So, and this is Xenexton, which is an amulet to ward off plague. It's based on Paracelsus's ideas of a future amulet to ward off. Like they used to have broad spectrum amulets to ward off disease. They would just ward off disease in general. But he thought for plague we really need a specialist thing. And so there's a debate over 100 years over what this should contain: sapphires, menstrual blood gathered on a full moon, toad skin, things like that, which is all incorporated in this 3D printed amulet. But this one really does ward off plague, because I collaborated with Professor Christine Rollier, who's from the Oxford Vaccine Group and now Professor of Vaccinology at Surrey. It contains the ChAdOx1 plague vaccine, so it really works. Working with Alex May, we've made sculptures that are along Brighton Beach at the moment, which are called the Black Rock Beachcombers. And these are digital collages made with photogrammetry, all different aspects of the beach. Also mentioning pioneering women scientists. These seaweed here are from a Victorian seaweed collector who lived in Brighton called Mary Philadelphia Merrifield, and we went and 3D scanned her collection in the Booth Museum, and now they're immortalised back on the beach where she collected them. The parasols from the ladies who walked along, and I know at least one person in the audience will recognise bits of the Pioneer, the Volks railway that used to go along in the sea, and the Rampion wind farm, and all sorts of things, all collaged together. So they all work with augmented reality as well. Other projects involve CRISPR modifying yeast to make a yeast that can capture CO2 and output lactic acid. So it's modified, so it feeds off CO2, and what it excretes is lactic acid, which you can polymerize to make polylactic acid, which is PLA, 3D printing filament. So you can make as much plastic as you like from the CO2 in the atmosphere that we're trying to get rid of. So it's making a kind of comment on the possibility of biotech in the future. Okay, it doesn't work that well. This little blob here is the PLA we actually managed to make from it, but you know, it's nodding towards that direction of what will this stuff do in the future, and is it wrong to say we shouldn't modify things when we could come up with new solutions? It's about questioning this. And another piece there is called wood spirit amber acid, and it's also about how yeast can produce different useful chemicals. Made underwater robots with Alex May, and this is Archaeobot, a post-singularity, post-climate change life form that it likes to live in very highly acidic conditions. It is based on archaea, the most ancient forms of life on the planet, how they swim around with their tails. But we were thinking about future fears of people, so climate change and also AI. This robotic archaea has a neural net, and it's learning about its environment as it's there in front of you. And it's currently on show at Diriyah Art Futures in Riyadh at the moment, which some of you might know Irini Papadumitriu, who used to work for Future Everything, who's now leading that organisation. Other works I've done have explored tuberculosis. I'm kind of obsessed with that, from interactive installations to works containing tuberculosis DNA or exploring the genomics and healthcare treatments. And one of my most recent works with Alex, we're collaborating, is creating an expanded data set, working with scientists who are using artificial intelligence for infection prevention. So the system they're creating will say, well, Mr. Smith in bed 4 needs to be moved to a side room because the AI says in 4 days' time he will get MRSA infection. He hasn't got it now, but based on all the data that we have about where he's been, who he's been near, what procedure he has, he's going to get this. So scientists approached us to work with them on this because they want to talk to people about things like how do people feel about giving up their data of how they move through hospitals to train an AI that might help them stop getting infections in hospitals up front. So we thought, well, this is the future of epidemiology. So we looked back. What was the start of epidemiology? Well, I knew very well, but it was John Snow mythically removing the pump handle from the pump in Broad Street in London to stop the cholera outbreak there. And he has an amazing data set for the time. 616 people died, and he made a Voronoi map. But we wanted to expand that data set to something that the AI now could learn from. So we're using generative AI and vibe coding techniques as well to create characters, people, backstories of all the people based on what we know about the people that were affected. Things like baby Lewis, who was patient 0, that's there. And using vibe coding over the map to create ways of following how people moved around the pump and things. And what we're going to do is test their AI on our expanded data set and see if they can stop the Broad Street pump outbreak from like 1854 before it takes hold. So that's where we're kind of are. And this is the cholera dress that I made, working with Wellcome Sanger Institute, where I'm an artist in residence. And that dress is impregnated with the world's oldest living cholera, from 1916, and it's antibiotic resistant, which is interesting because it's before the antibiotic age. So it shows that resistance was there in the environment before we have the antibiotic resistance that's caused by overuse of drugs now. And I had to get a little story. I had to get Schedule 5 authorisation under the UK Bioterrorism Act to do that work. So I'm always like having to do lots of like forms and things like that to try and be allowed to make the work. But I'm quite tenacious and I don't let the forms stop me. So that's the sort of background of what I've done up to this point. And then I wanted to talk about, I did a residency. I got asked to do it in the auspicious time of February 2020. So I ended up doing a 1-year kind of online residency with the Helmholtz Zentrum at the Institute of Epigenetics and Stem Cells in Munich in Germany. And then as soon as I could, when it first opened up, I was able to go there with all these tests and paperwork and test to move again. It was terrible, absolutely terrible, but we got there, and I started to do experiments with them, and everything was starting. So they were like, we need an artwork in 3 weeks time to show at the what's it called, the Deutsches Museum in Munich. So I was like, no pressure, okay. So we started to do experiments in the lab. And I learned about this thing called chromatin, because we all know about DNA, it's a double helix, but the DNA is wrapped around these things called histones, and whether they're tightly together or separate is really what makes them different. So every cell in the human body has the same DNA, but as you know, some of our cells are skin, some of our cells are lungs, some of our cells are liver, some of our cells are heart cells, all sorts of things. It's the epigenetics, the way that this chromatin is either tightly bound together or separate, and can be modified in different ways, that makes the cell what it is. And that's what epigenetics is. And this is a zygote that we looked at under the microscope. And that's the mother cell, and that's the father cell, and the yellow is the chromatin. And it's before they fuse. So it's just been fertilised, but the egg hasn't, the two cells haven't fused together yet. And so I made this necklace based on chromatin. And so this is the chromatin structure, anatomically correct, of the OCT4 gene. What's that? It's a Yamanaka factor. So this is something that you can use to make a cell into a stem cell. So all stem cells have the OCT4 gene, and they don't if they're not a stem cell. And a stem cell, these induced pluripotent stem cells, they can become almost any cell. So you can apply different growth factors to them, and they can become almost any cell. There's something beyond that called a totipotent cell, and that can become any kind of cell. So what's the one thing that an induced pluripotent cell can't be? It can't be placenta. That's the sort of, if you can get the most perfect cell that can become anything, it can also become placenta. So this is it's called the cellular reprogramming necklace, and it's anatomically correct representation of the OCT4 gene. And these are the H3K9 trimethylation bits. I'm going to use techie words, but it doesn't matter. It's 3D printed. And it's really hard to make because in the natural world, DNA wraps magnetically around the histones. But in the real world, you have to attach the histone, which is a 3D printed thing, to the bit of material. And then you have to drag it right to the end. And then you have to turn it round. And then you have to do it with another one right to the end. So you're not like just wrapping it around like the magnets would do. So it represents the heterochromatin and euchromatin, the far apart and the tightly knitted together chromatin structure of how the DNA is. And this is the thing that they call cell fates. So cells, do the cells know what they're going to be? How do you make the cell into a certain cell type? And if you wipe the memory of a cell, does it forget what it used to be? Does it have a memory? It's called cellular memory. So I came up, and this is a long. So this is the thing I showed at the Deutsches Museum after 3 weeks. I made a few versions of this now, and they're circulating around the world. So that's good. It went well. And it's got copper sulfate on it, and we were talking about this earlier, because in the experiment, I'm always looking for the materials that they're working with or things that I can incorporate in the art. And they added, in the experiment, we just add a little bit of copper sulfate to the cells. Why? Oh, they need it to live. So it was like, I could use that. So I came up over about a year or maybe 2, and we ended up extending the project for another whole couple of years. I came up with a question, and I've got a little tiny video to show you what happened with the question. So I'll just go on to that. So there's this question, do cells have memory? Do they remember the cells they were before? And I said, well, maybe we could try and see if they did. Like, why don't we get some stem cells and make them into like one cell type, and then take them back to stem cells and then turn them into something else? But what if we could do that 4 times and then we'd know?
This was quite surprising because I realized that no scientist had actually thought about that before. So we had to wait until Anna came and asked her, we can change the fate. Theoretically, we can go back and forth, right, as many times as possible. But this has actually not been addressed. And so there was a significant amount of time spent on trying to see, can we actually do it?
This is actually a very romantic conception of your research topic. This kind of poetic questions also have other implications and other layers of meaning. And this is actually where you also always are very strong in bringing together these different ideas. You're taking some of these beautiful imagery, you're taking some of the very specific scientific information that the scientists provide to you, but you also engage yourself in projects directly in the lab, and you also try then to go even further and connect to cultural history and to everyday life aspects, so that in the artwork you have all these different layers.
That if you think about your own body, you have all these different cell types, right? Your skin cells will protect you from injuries, your heart will beat and make you having the blood. But they all come from one single cell, which is basically the result of the egg and the sperm at fertilization. And that cell has the same genetic material. So the DNA is exactly the same throughout all your cells, with some exceptions. And so for me, the question is, how on earth can we make all these different cells that make our body so complete and so complex from one single genetic information?
So the cells have been stem cells, then neuronal progenitor cells, which is basically brain cells, back to stem cells, then to heart cells that actually beat in culture. What we noticed about those is they beat irregularly. This happened when we were doing the soundtrack. We don't know why. So it's another question that we came up with today. And then they go to back to stem cells, then to hepatocytes, which are liver cells, and then back to stem cells, then to lung cells, and finally back to stem cells. And I'd like to turn them into totipotent cells that can become anything, but the research hasn't caught up with my artistic desires yet. So that was Maria Elena Torres Padilla and Claudia Schnug, the curator. Maria Elena is the director of the Institute for Epigenetics and Stem Cells. And so so this was the work, and then we also showed alongside it, so this was a video of the cells, we showed alongside it a bottle, like a genie bottle, that contained the cells that had been the 4 different things there. And there's also a frozen set in the lab at the Helmholtz Centrum that could be woken up and made into totipotent cells one day, if that becomes possible. And these cells... The scientist I worked with on it said that he thought they got faster at transforming. They didn't know if this would ever be possible for cells to go back and forth and live kind of 4 different lives, but it was. So, there's a close-up there. I asked the question, were eggs totipotent cells? And that's a big philosophical, weird question, so we don't have time to go into it now. And that's a close-up of the bottle. I also worked with the Gurdon Institute on this project, the Gurdon Institute in Cambridge, who are pioneers of IVF technology, and they work a lot with donated embryos to do their research. And so I made this piece, Precious Cells, which you can find on my website, and there's an interactive version actually on the website. But they have cells up to sort of 7 days that they study, human embryos. And this period is very important, like the beginning when all this is happening is really important to humans. And so next slide's a bit of it's not a piece of my work, it's a piece of history, but it's a little bit more traumatizing, so trigger warning. I'm going to show an image of the Dutch hunger winter, because I got into the question of like, well, if I was epigenetically different, what would I look like if I'd been in a different situation and stuff like that? If I was, there's this idea of twins, if they had brought up in a different setting or something like that. And there is one scientific kind of proof of this, which is the Dutch hunger winter cohort. And so in 1944, the winter and early spring 1945, there was this period known as the Dutch hunger winter when the Nazis blockaded the Netherlands because the Nazis were trying to help the English allies. So there was a rail strike, and so they prevented them from getting any food for like 3 months, especially in northern Netherlands. And so people were actually starving. At least 18,000 Dutch people died of famine, and they ate all sorts of things. Famously, they ate tulip bulbs, which are now a delicacy. You can get them and eat them, but they have to be organic and not treated with any chemicals. So some very high-end restaurants in the Netherlands will sell them, but they couldn't export them because of the blockade, and they didn't have anything else to eat. They ate cats, they ate dogs, they ate chicory root, which they do grow there a lot. Got a picture of some chicory. Did a project on chicory as well. Don't have time to talk about it, but it's on my website. Also did they ate fava beans, the tiny horse beans. Also have a project about that on my website, but don't have time to talk about it. Don't know why I know so much about the Dutch diet. I'm not Dutch, but over the years I've done all these different collaborations, and so the women that were pregnant in the Dutch hunger winter, whose babies were in the first trimester, because it's a very discreet, like, separate thing. The Dutch people were very well documented beforehand. They knew who they were, where they were, and they had loads of data. And afterwards, again, very well documented. They knew who had died and things like that. So they were able to find the women who were pregnant in the first trimester during the Dutch hunger winter. And so later they went and tracked down the babies of those, adults now, elderly now, of those, if still alive, of those women. And they found out that they are upregulated for diabetes. So they're much more likely to have diabetes, and they have a much slower metabolism. So even though they were about that big during that famine, their body was formed in a way that it was set up to live in a world of famine. So they were ideally suited for a world of famine, but then they weren't born into a world of famine. They were born back into the everyday world that we're used to. But so this physical trauma is imprinted in their cells. And then Operation Manna happened, which is the title of the project. That's why it's called this. It means food of the gods, but it's these Lancaster bombers from England came and broke the famine. It was a very dangerous thing because the Nazis said they would have a ceasefire, but because they were already really close to losing the war. But they didn't know if they wouldn't shoot them down. So they dropped these food parcels there. So it was really interesting. My mum actually painted a picture in the wall. She was evacuated because we were talking about as well, like our stories about this. My mum won a competition for painting a Lancaster bomber when she was evacuated in World War II. She's dead now, but she told me that story. And so there's all these things, these like stories that are passed down. So I did for the prototype of the project, I also did a workshop I had a show. That show that you saw was in Hallein in near Salzburg in Austria, a place called Altsaline, an old salt. Well, it could be in Salzburg. It was an old salt processing plant where they have the Schmiede festival. And I did a workshop for that Schmiede festival, and we made fetuses. We 3D printed them and cast them, and we made biomaterials using the foods that were typically eaten during the Dutch hunger winter. So smashed up tulips and chicory root and chicory and all sorts of things mixed into this. And I was working on, and you saw probably a little spoon in the thing, because the children in the Dutch hunger winter said that they always carried a spoon in case they found someone serving up some soup. So the spoon became like a really important symbol. And so I went back I've talked to Cécile and Rachel, and we developed all these ideas together, especially around things like intergenerational trauma and post-memory, which Rachel will speak more about. And I went back to the at the Helmholtz Centrum, and they were able to create for me the OCT4, not the OCT4 gene, this is the PIM3 gene, which is the gene that's affected. So it's actually had the DNA, I got the DNA of the gene that's affected in the Dutch hunger winter. And what I created was this 3D printed monstrous plant, if you like. That's a close-up of it, but that's what it looks like. So it's 3D printed. It's a tulip, but it's also spoons there, and it's chicory and sugar beet. They ate whole sugar beet. They could get that. The Dutch produce a lot of it. The tulip, the fava beans, some small potatoes. It's a chicory root and a sugar beet root all collaged together. And so it's a similar techniques that I used in the Black Rock beachcombers with Alex that we've used in this. And so you can see at the centre of the tulip bulb is the tiny fetus of the age that was affected. And that's impregnated with that OCT4 gene's DNA in the artwork. And it's currently on show at the Science Gallery Bengaluru in Bangalore, in India, where I also did a workshop where we talked about famine there as well. So Rachel spoke to me a lot about famine as a weapon of war. And so also during the colonial period, they had the Bengal famine in India, but it's very hard to track the direct things. The Dutch people were completely healthy and well-fed before and then again after, so it was a very short period. But there's been lots of sporadic famines in different places in India, but they do also have this very high rates of diabetes there, so it's a similar thing. So these sorts of things have caused it. So the Bengal famine, they also had a cloth famine and things like that. So people there were very interested in the artwork to tell this story. It's part of an exhibition called Calorie, which is all about food. And so I did a workshop there, and we made the same little models, but cast the same models, but we used things like banana stems, because that's what people in India would eat in a time of famine, because the rich people get the bananas, the farmers are just left with the stems. But apparently they're now a delicacy, so, you know, it becomes fashionable. So what I'd like to do now, I've got a little, let's see if you want to keep in touch, you can take a slide of that, take a photo of that. But I also have cards, and I'll just put a picture of that up, and I invite Rachel and Cécile to join me, and thank you. Yes. So in the middle. So, yeah, do you want to start? And you were going to say a bit of feedback on your experience of the project and.
Yeah, okay. Yeah, do you want me to start?
If you like.
Yeah. So this has been it's been a great collaboration to work on and to work with Anna and Cécile on this project, and I've learned so much from it, both about epigenetics, which I knew nothing about to start with, but also from Anna's artistic practice and the kinds of things that she's looking into. So I sort of came at this, I'm not a scientist, I'm a historian, contemporary historian, and I'm in the Department of War Studies. And my research and teaching for years has been on, sort of very broadly on consequences of war, if you like. So how individuals, communities, societies deal with conflict and its aftermath. And for quite a long time, I've worked on transitional justice or post-conflict justice, and looking at international criminal tribunals, the ICC, and that kind of that area of response to violence and past violence, how it's contended with in that way, and how people are seeking justice and accountability. And in doing that, sort of also looking at and investigating the shortcomings of that kind of approach, particularly for survivors of violence, and particularly of atrocity, who hold trauma and are looking for not only justice and accountability, that's certainly there, but also recognition, repair, in some cases reconciliation. And so we started looking at the project, and this is sort of comes around to being relevant to this. I did a project of starting a few years ago, looking at art and reconciliation, and this question of, well, what's the potential for arts and creative practice in processes of reconciliation? And we started looking at it in perhaps a bit of an instrumental way, kind of looking at, well, what can it do? This question, what can art do? But one of the things that was most transformative for me anyway in the project was working with artists and curators and museums as a methodology. So really thinking about, well, what not only what is art doing instrumentally in practice of reconciliation, how is it being used by community groups and participatory projects, but also what can it tell us, what was working with these different groups of people and working with artists particularly telling us about the questions that we were asking, and the sort of the way that we were going about investigating, and the kind of the conceptualization of reconciliation in the first place shifted. So that was a sort of window into I suppose the benefit for me as a researcher of collaboration, and of collaboration not only sort of interdisciplinary collaboration with other scholars in international relations or in across different disciplines, but these sort of artist-researcher collaborations. And then I was just really fortunate that I was sort of sitting there, and Cécile, who I know because she's a curator and part of war studies, had met Anna at a sort of, I guess it was, I don't want to say it was a speed dating event, but it was a kind of science culture
art sanctuary project
program,
organised by King's College.
Yeah. So King's have a section called King's Culture, and they had this program of seed funding around a program of work called Sanctuary, which ended up being called Lost and Found, I think. But they were bringing together artists who were interested in this theme and exploring this theme, and interested in working with academics at King's on this sort of broad theme of sanctuary with academics. So Cécile met Anna at this event, and then introduced Anna to me as well, because I work on sort of post-conflict trauma and consequences, and she thought it would be interesting and have some intersections, and it was, and it did, and it certainly made me think quite differently about what I was working on and how I was thinking about consequences and this sort of particularly how individuals, but also it has sort of implications for thinking about societies and communities in the way that they deal with conflict and its aftermath, and what it leaves, what it leaves in the body, but also kind of translating that into what it leaves in society and in sort of socio-cultural aspects as well. So I think Anna's already kind of described the project and the process, and so I thought I'd just focus briefly on the contribution that I think it made to understanding from my perspective, way of sort of collaborating. And as Anna said, we had sort of series of conversations around the Dutch winter famine, around sort of the broader issues around famine as a weapon of war, and we were talking about the International Criminal Court indictments for use of starvation as a weapon of war in Gaza, and the sort of prevalence of famine and of starvation as a weapon that's used directly against civilians, and this sort of being an illustration of that, and being a really stark illustration of the kind of intergenerational harm, which was sort of where we went with it, where you started with it with the epigenetics. And I think when you were talking, I've written down layers, because when in our conversations, I felt that there were constantly layers and layers and layers of meaning, and so the collaboration itself, when the three of us were talking, there was always sort of sparks and something else that would be relevant that you bring up. but I think I've sort of distilled it down into, I suppose, three main sort of arcs of significance that I wanted to focus on. So the first is this is the significance of the sort of biological or the physical effect of trauma on the body, and the way in which these events and experiences are imprinted on us, which I think is illustrated in the epigenetics. And I'm not a scientist, and I know that there's some some of this isn't is contested, isn't it? And but I think from coming at it and just take it, I think, I suppose what I took from it is the kind of headlines from it that seem really significant and resonate with what we know about and what we're interested in investigating about the effects of trauma and conflict on people. So this sort of one aspect of the physical effects of trauma on the body, and how injury, even if it's invisible, so this idea of sort of the invisibility of the impact, can be transmitted through generations. So the research on the Dutch famine has resonance too with other instances. There's been research too on intergenerational transmission of trauma through Holocaust survivors and into subsequent generations. A bit of research about Rwanda and children who were born from mothers who were alive during the genocide in Rwanda, show us this sort of potential for transmission through not only one generation but two generations. So that was sort of the first sort of starting point of it, was the kind of physical and the bodily impact, bodily injury. And the second was the significance for understanding the effects of famine generally. There are many examples historically to think about, as Anna's already talked about. I think there was 61 famines recorded since 1870, and it's extremely prevalent now. So Alex Duval, who works on Sudan and other places, had said that there are there's 363 million people worldwide currently facing starvation, and we've seen this, I suppose, most well reported in Ukraine, which obviously also Ukraine's really interesting because of the kind of reverberations of memory back to the Holodomor too within Ukraine, and sort of I think that sort of ties into these ideas about kind of reverberations of how memory is transmitted and how injury is transmitted and comes back again. So Ukraine and in Gaza and in Sudan and elsewhere. So it's really significant to draw attention to it and to use art as a way of opening up conversations about the effects of conflict, and particularly about the strategy or tactic of using starvation as a weapon of war. And then the third aspect, as Anne mentioned, was the way it resonated with the research on psychosocial or psychocultural effects of trauma. And intergenerational transmission. And I think when we first started talking, the first thing that sprung to my mind was Marianne Hirsch's ideas about post-memory, concept of post-memory, which is concepts that tells us about how traumatic experience is not only passed through generations, but can be felt even more strongly by the so-called 1.5 generation or the subsequent generations than the immediate one. And Hirsch showed how this could operate via visual and cultural cues. So that really resonated for me with this idea of epigenetics and the kind of the tags. We talked a lot about tags turning the genes on and off in response to stimuli. So with post-memory, intergenerational or transgenerational memory is turned on and off in response to visual cues that are presented, and it's these intergenerational acts of transfer that make this embodied or living connection between the past and the present. So what do we take from all of this? I think there's a couple of things. One is that traumatic experience appears to be transmittable through generations, but that transmission isn't linear, nor is it certain. So it can be one thing that I sort of kept coming back to with this was the idea that the things that we do to try and mitigate trauma can end up being the things that make it worse, as a sort of theme to explore. It can be either mitigated or worsened by our actions, and that the transmission and response are mediated. So it's always mediated by cues. So it might be mediated by what happens to people post famine and what happens down the generations. And the concept of post memory, I think, helps us see that and understand that how things aren't necessarily preordained. The potential is there, and then that it's turned on or off by these visual cues, by what we see or what we choose to see. So I'll stop there and maybe hand over to Cécile to
Well, I think you gave a fantastic context to the reason why we work with artists is exactly this idea of giving shape, giving form to all these ideas and concepts that you are just elaborated. And I'm fascinated by the work of Anna, because she's a true believer in material. She wants to fuse as well history with legends and the present, and that capacity to encapsulate all these dimensions is really an artistic work. It's an act that you're posing each time. It's each time, you revisit our knowledge, science, and how the imagination out of these, I would say, precepts and cells can bring another dimension. So I think this is one of the reasons why I'm always convinced to work with artists, is because they surprise us with ideas, with encounter that we would never ever imagine ourselves, and also to bring within that storytelling, because Anna, you're a fantastic storyteller. I mean, come on, this is absolutely amazing. How to bring those subjects that are so much, part of our present time, as you were mentioning, Rachel. Starvation is more than just a weapon. It's just such an inconceivable, strategy when we think about it. And I think Anna is just bringing that perception of it through this past memory question, and how we are all embedded in it, how we are all can be becoming actor of a knowledge that has been passed through our bodies, and that has often been, I would say, not transmitted to many generations. And the fact that you went to Belaguru and spoke about the Bengal famine is extremely important, because it questions colonial time and what has happened during that time. And not only in India, in Bangalore, but in many other places in the world. So here I would like as well to refer to this capacity you have to speak about this belief in materiality, in material, is super interesting, related to how we question science, how we question the capacity to bring memory, and fermentation is a fantastic, I would say, tool for thinking about all that. But beside that, I think what was at the beginning of your presentation, and that struck me when I came to your studio, was how you've been working on
Epidemics.
How
Infection.
epidemics and infection are bringing this notion of fear, like embedded fear in our bodies, in our mind, in our imagination, and how you speak so lightly about it. It's fantastic, because I think we're just being kind of traumatized as well with COVID, and how it speaks to us nowadays is quite remarkable, because through this work you bring us to our own understanding, our own fears. And I think this is what is strong with this work. And I would like as well to speak about the chromatin necklace, and that we've seen earlier. This is remarkable, and this project that has been presented in Belaguru, and that we're also going to present hopefully at King's, is also extremely interesting, because it's a fusion of history and the practice of famine, how people were resisting. So this work is also very hard. For the first time, this work is quite, to me, it resonates as well, because it has a very sculptural effect. I cannot stop thinking about Giacometti and how himself as well has been talking about, for example, epidemics and famine. And how this works has a very sculptural effect, although it's quite small, right? It's 60 centimeters large. So, and then how you bring back that memory of the spoon, right? That spoon we've seen in those black and white pictures of these toddlers in Holland. So you really create this kind of fusion of all these elements of existence, and I think this is why I think your work is very important, because it is really at the crossroad between research and art, and this is exactly what we're interested in, working with artists as well in water studies, how artists are researchers, and Anna proves it by themselves in their own means. And I think this is really important, to keep this possibility of thinking how to visualize trauma, and how artists are playing a big role about this. So thank you.
Thank you.
Thank you.
I was going to say, one of the things that was really interesting in Bengaluru was that when I did this workshop, one person that came up to me, it was mostly quite young people in the workshop, like in their teens to 30s, let's say, and one came up and it was exactly this post-memory thing, because he said that he realised that he was really angry about the Bengal famine, and he said, my grandparents who lived through it, they're not as angry as I am. I'm really angry about this, and I haven't really thought about it in this way. And because of this artwork, and what I was told by the Science Gallery Bengaluru was that it's really good to have this, like, it's this story from the Netherlands, but to present it in Bengaluru gives them a way to be able to engage with those issues without having this, like, oh, it's another thing that's happened to India thing. It's like it was like showing different cultures, also like the kind of cultures that are maybe presented to be the rich West culture, but it's a way of enabling them to discuss it as well. And the other thing I forgot to mention in my presentation was the three generations thing, was because I forgot to mention that if it was if the embryo was a girl, it contains the eggs of the next generation. So the epigenetic effects are definitely passed down to at least a third generation, so on. So
when we were talking last week, getting ready for this, I think, Rachel, you also mentioned the idea of the object and that providing a form of distancing. Mm-hmm. And I was in my mind, because obviously you're creating these works, Anna, for people to look at perhaps in a vitrine or establishing some kind of objective frame around it. I'm quite interested in that process and why it works and why you think it might work, and in the design of the actual display. If you had any thoughts on that?
I mean, I'm a sort of practical person. It's a very busy gallery, so they wanted to have a vitrine around it. And when I showed it in Halline, the spoon version, the earlier version, I showed it open. So if it can be, like if it's in that kind of space, it can be open. But if it's in a place where you're going to get 100,000, 200,000 people coming through, because it's really busy gallery and it'll be on for a long time, they protect the work like that. So it wasn't necessarily an aspect of the like my intention to have the big case around it. It's just that you might need somebody on it all the time because it's quite fragile. I mean, there might be deeper things to say, but I'm always quite a practical person. So
on the same note, planning something for kings, for example, and where it's going to be placed, how important is it, the communication strategy that is evolved around an object like this, and how do you go about making that work?
I think we have to give a context, a background, because not everybody is aware of, the Dutch famine. As you did in India, I mean, it needs really some kind of information next to the work. Yeah, that's really crucial.
Yeah, we'll write it collaboratively. I usually like writing text. For exhibitions, I usually write it collaboratively with the curators and collaborators. So we'll write it like that, aimed at the audience, which will probably be your students and the sorts of people that walk through King's. So aimed at a general audience to introduce them. But I try to do it where I don't have like masses of text. I always need a bit, I think. You won't know what I've done. Like pretty much all my work is you won't know what it is unless there's at least a bit of a label, I think.
Yeah, I think you call it stories behind the stories in one of your interviews, which I think is a great description. Do you think it would be appropriate to open up for other questions now? Perhaps you could just see what hands go up rather than me monitoring them. So
there's a few. Yeah.
I was just going to say, I think this talk about on epigenetics is really interesting. I've worked with 2 birth cohorts in the UK. I used to work for the 1946 cohort, where they were post-war, one of the oldest cohorts in the world. Babies born in 1 week in March 1946, and there was about 5,000 of them. They tracked them through entire lives. It was meant ostensibly to be for health, but the questions and the questionnaires themselves reflect what was important in the post-war thing. And we had nutritionalists and people working on there, the scientists, and they were saying, for that cohort, they were on rations for 20-something years or many years. And the question arose of whether the health trajectory of those people, echoing the recovering from the war, not famine like the Dutch, but famine kind of lean years.
Yeah.
Whether that would be representative of, say, the 1958 cohort, or the 1970 cohort, or the ALSPAC 1990s cohort, in terms of that memory of you grew up lean, and what impact is that going to have when they have kids, and that. So it's it might capture some really interesting just post-war memory, because they were interviewing the mother for years, and then they interviewed the kid, and they're still alive, well, some of them anyway.
Yeah, that sounds like a fascinating data set. All history maybe combined with the epidemiological history, whatever we say, genetic. My mum was born in March 1946.
Interesting.
She was part of the study, but
you see the old yellow typing paper where they were literally typing out the questions and getting the answers, and then you've got the punch cards. You've got these old microfiche things and the reels. They went through every era of computing, and they're probably one of the most questioned groups in the world. And they probably have that memory of growing up, at least even now. They might be really interesting for you to go and check out.
Okay. Thank you. I think there's one question there, and then come.
This is not a scientific question. I realized if your institute is studying war, then that's what you do. But when I was listening to you, Anna, I was amazed. It was like such a joyful presentation, and And what strikes me is the whole spectrum of things you could study. And this seems to be about war and famine. But life itself is ecstasy. You know, you may disagree, but it's full of like the sun in the sky, giant amounts of energy. All these plants and animals are springing into life, you know. I was born in 1942, and I remember being alive was ecstasy, you know. I mean, the raindrops on the windows, the light, everything, my dreams. And this is, how can I put it? We seem to be obsessed with dealing with the dark. And what you're dealing with now is how to be fundamentally creative, you know. Because very soon we're going to have artificial intelligence which can turn on like the sun, if you like, and wipe us all out in 30 minutes if we're not ready for, you know. Anyway, it's just a statement. I just wanted to say something.
Okay, thank you. I mean, I guess I'm just fundamentally fascinated in the weirdness of everything, and when I find these like strange effects that have happened throughout history and are happening and will happen in the future, I'm really fascinated in sharing those stories and exploring them more and exploring them hands-on and going into labs and talking to historians and just developing those ideas. So I find it very fascinating, and I'm just like, you know, I'm building ideas. It was when I met Cécile, we were talking, and I was like, actually, several of my works do relate to war, and I have this idea for a project, and we started talking, and that's how that evolved. And I guess I didn't also expect that I would be doing a collaboration with War and Department of War and Society, but I am. I didn't expect to be doing a project with human fecal transplants, but I've done that too. So you know that, but these things are so fascinating and so important for the world, like infection, war, and how all these things intermingle affects us all. So and we can't deny that. We just need to think about and engage it. Otherwise, the world's just really terrifying if we don't think about it. When we learn more, it's like maybe there are solutions.
Yeah. And I think we need to find ways of thinking about these very dark, difficult, horrible issues in creatively. And I mean, the collaboration has been fun, hasn't it? It's been, you know, the conversations that we've had are really been really really rewarding and really kind of generating. I don't know, you know, kind of invigorating in a way, because there's so much curiosity there. I suppose we all just got so much curiosity about these issues, and talking to each other and having the sort of creative aspect to it sparks different ideas. So it's been a fun collaboration on it. I mean, the topics are difficult topics.
And again, it speaks about what is happening in the world today. You know, famine is present. It's really, you know, yeah, you gave numbers, but at the end, it's just human beings after human beings starving. So this is what we're talking about.
There's a very interesting YouTube video. If you look up Dutch Hunger Winter, I didn't have time to show it tonight, but it's like it's maybe a 10-minute video of a guy who's found all the footage of people queuing up and the children looking for the food and things like that. And it's just a short video about it, but it's really fascinating if you get a chance to look at that.
Had a question?
Yeah. And thank you for your question statement. And thanks for your presentation on it. It's really interesting. It's had me thinking about, I suppose my question is around the artist research process and the scientist research process, because Cécile, yes, you're right, artists are researchers too, but in very different ways with very different end objectives, if there even is an objective. And I loved hearing about the making plastic out of thin air. Beautiful. And I wondered if maybe you could speak about that as an example, or if you have another process with a scientist in mind that you could share as an example of the way that you worked and or maybe crossed over, or is that a sharp question?
I'm trying to think of a good one. So a piece I didn't put in the talk because it's too complicated to explain briefly, but now you've given me an in, so I'm going to do it. There's a piece called Make, Do and Mend, which is based on this idea from 1943 during the war that we should patch and repair things instead of just throwing them away. Now, 1941 is the start of the antibiotic age. It's the year that Florian Chain first trialled penicillin in human patients. So I was working with the Modernising Medical Microbiology project at Oxford, and how I first got in with them, and I've been working with them ever since actually, so from 2010 I've been working with them, was my collaborator before that, because I managed to meet a scientist, I got introduced to a scientist, started to work with them. He was it's called regional microbiologist for London and the South East. So he's like in charge, like if there was a flu epidemic or something, he'd be in charge of how that was coordinated. And but when I first started working with him, he was just a consultant in a hospital, like the consultant microbiologist in the hospital in Brighton. And he ended up going to a meeting with Modernising Medical Microbiology, and they were like, well, we've got this big grant now to look at how genomics can improve infection prevention, because we can diagnose things, we could look at whether they're antibiotic resistant, we could do all this. We've got this grant now, but I suppose we better do some public engagement because that's part of it. And he was like, well, I've got an artist in residence. And they were like, that's great, bring her down. So I started working with them and started working with like genomics around MRSA, bacteria, tuberculosis. They decided to work with the 4 like big hit, like famous ones in the press. So it was C. diff, and they worked with norovirus as well. But I didn't really do much with norovirus because I don't do viruses. I like bacteria. And so then I started talking to. I had this idea because we whole genome sequence the Staph aureus from my body. I alluded to that earlier. It lives in my nose, in the front of the nose. Staph aureus, which is also can be methicillin resistant, lives in the front of your nose because it can stay away from when you get a fever to kill the bacteria in your body. It's cooler there, so it hides in the front of your nose. And we found it was penicillin resistant, so that was exciting. I was like, oh, that's nice. I've got a penicillin resistant Staphylococcus aureus living in my nose. We don't know if it's always penicillin resistant, might be for some time, might be methicillin resistant for some time. It can change. And so I had this idea to remove the antibiotic resistance gene and take my Staph aureus back to the pre-antibiotic age, back to before Florian Chain did that trial. So over several years after this, the science started to catch up with me, and it became possible to CRISPR modify the bacteria. Well, I didn't actually manage to do the bacteria from my body. Still would like to do that. Anybody watching online, scientists? But I got a bacterium that had a penicillin resistance gene, and I used CRISPR DNA modification to cut out that gene and repair it with the phrase. You have to, bacteria genomes are kind of circular, they're all twisted up, but they're sort of circular. And so if you don't actually put in a repair fragment, it just flops apart and dies. So you have to write a repair fragment. So in DNA, I wrote the phrase make, do and mend. from ASCII code into base 4 into the DNA bases, made a repair fragment for the DNA, edited out the penicillin resistance gene and repaired it with the phrase make, do and mend in the DNA of the bacteria, which then I grew onto clothes that were stamped with what was called the utility mark from World War II, which the utility mark meant it was like kind of good for the rationing that used stuff sustainably. But it was stamped, it looked like Pac-Man, CC41, which stood for Controlled Commodity 1941. So I thought this is fascinating because antibiotics were not a controlled commodity. We've used them willy-nilly, and we've got this situation of antibiotic resistance genes. So managed to do that and then grew it into silk and then used it to actually patch and repair, like in the make, do and mend idea. So could we make, do and could we mend antibiotic resistance using CRISPR DNA modification, which is this relatively cutting edge, it's been around for a bit now, DNA modification technique. And so I made the collaborations to do that. I had to travel to different parts of the world to do it and things, working with different cultures as well. I worked in the UK with it, and then exhibited it in Oxford History of Science Museum originally, which is where Florian Chain's collection is, and they have their amazing things, which are these like their fermentation, they fermented penicillin in biscuit tins with tin cans welded to the side because the war was on, they couldn't get like any instruments or stuff made professionally, so they literally biohacked these things. So you've got these kind of biscuit tin or cake tins with things stuck on, and they use lidded bed pans to grow the bacteria, grow the penicillin as well, which was then terrible when they used it in people. It burnt, and they used these big needles, and it was hot, and then they would take the urine from the patients and go and re-extract the penicillin and then give it back to them and stuff. So that's sort of how it goes. I don't know if that answers your question. We published it in Leonardo, and I managed to show it in the CDC Museum, the Centers for Disease Control Museum in America as well. So then it goes off and it has its own life going around and try and develop things. That's sort of my process, but I don't know how that counts as really, I mean, it sort of counts, it's kind of artistic research and it's my process, but like it's quite hard to pin me down into a research process. I think.
Because you've been working with people who are making wine as much as, so many different levels. You've been touching many different practices.
Because I'm very much based in, although I work with a lot of academia, I'm very much based in like the, I guess, art side of things.
I suppose my question, I didn't phrase my question correctly. It was, I'm curious about when you're working and sharing information with scientists, you're sharing knowledge, you're developing your knowledge that you offer to the world, and they're developing their knowledge, and I'm curious about where that intersects. And your end result is very different from, perhaps it's very different from theirs, or perhaps there is this dual process of, it's play and experimentation and things going wrong that something emerges from. And I imagine there's quite a similar process between an artist and a scientist in some stages of the research.
Oh, yeah. And I'm doing the lab work hands-on with them as well. So we're all learning how that works together, because no one would ever remove an antibiotic resistance gene. You put them in to a thing to see whether your experiments worked. It's a completely backwards method of and it's really hard to see whether it's worked. It's much more complicated. So they'd never tried that. And why would they? It's like we're trying to get the cells to go backwards and forwards, what she was saying. Nobody'd ever tried that. They hadn't even thought of doing it apparently, but it seemed quite poetic as well, so it's like bringing the poetic side in.
Can I just ask a question? So you're obviously extremely passionate about many areas
we say in science, and you have the language for it. And I'm just interested to know to what extent you embed yourself in research or whatever before you maybe have the communication with a scientist. Do you feel that's part of your responsibility almost to have that engagement? To what extent are those scientists open to you coming in and perhaps being, on a journey of learning and understanding, or does it make it safer, if you like, for you to go in with a lot of knowledge?
It's a bit difficult to say from this point in my work because it builds. So I guess when I first started working with plant science, for example, I didn't really know anything about it. Somebody wrote to me and said, we've got these commissions to work with plant scientists on chicory plants. Do you want to do that? And so I went on a little journey doing a bit of research, and I found that chicory plant might have been Goethe's idea of the blue flower, which is the central symbol of the German Romantic movement, and that sent me off on a whole thing. And then I could talk to them, and then because I'd done CRISPR in this CRISPR DNA modification in other things, then I was like, I know how to do this in plants to an extent, but it's different. How is it different? And then you're learning with that. With the Helmholtz Zentrum, again, this was another of these closed calls where they take 5 artists and then they pick one. And luckily enough, I was picked. And that was really, so I didn't know much. I know about DNA. I know about DNA in bacteria, but I didn't really know about, and I'd done projects about even attaching fluorescent molecules to DNA and different things like that. But I hadn't done human cells really, or growing in these cell lines and things like that. Not much anyway. And so that was a very steep learning curve. Epigenetics is extremely complicated, and they work on very tiny little changes and stuff like that, and give very intense talks, lectures on these subjects. And I just sat through all their lectures, and because it was COVID, I had to have these 1 hour meetings with this guy, 1 hour meetings with this woman, over and over again through a whole year. I had loads of online meetings, and you feel like your brain's going to melt out your ears. But I kind of, over a while, it's a bit like being chucked into a country where you don't speak the language or something, and that after a while you start to
But that's the thing,
learn.
See, that I was thinking about that, about language. It seems to me that you learn the language, you're communicating with people in their language. To what extent are they actually communicating back to you in yours? It's, I think
they do. I think they do start to, because I think a lot of scientists have got quite an artistic side, or they're thinking creatively, or they play music and things like that. And so once they have it's almost, maybe it's a permission thing. Once they know that I'm safe to talk to and I'm not going to judge them or something like that, and it's a similar thing with me, then they're quite sharing of all that sort of side of things, and we just have fun usually.
Just wanted
It's
to
one
say,
question, I think.
sorry to interrupt as well, and then we mustn't forget questions online. Maybe
Okay.
Yeah, we had one question.
You had a question, no?
I think Jeff.
Can you hear, Anna?
Yep.
One question from Bernida Lights, which is, what precautions did you put in place for the plague dresses? Or dress that you meant?
So
the public concern about this.
Yeah, there was quite a funny discussion on the what's it called, the what's that famous? There's a famous museum in America, and they put it on, begins with S. Smithsonian, that's it. They posted an article about my work once, and the public were very concerned. But I can assure you, it's all safe. So DNA is actually sterile. So we actually, so at Colindale in London, with the National Collection of Type Cultures, we killed the plague bacteria first, and then we extracted the DNA from the killed plague. And I have a certificate letter from the National Collection of Type Cultures that I can provide that tells you it is dead, it's all sterile, and it's safe. So all my work is sterile and safe. And you can also refer you to an article I published in the Federation of European Microbiology Societies, FEMS Microbiology Letters, called Bacteria on Display, Can We, Should We, which I wrote with Nicola Fawcett, who's a researcher at Oxford University, which goes into the full nitty-gritty of the risks and benefits of showing it. But the gist of it is, the reason we do it, the reason I like to do it, I have this feeling, it's a bit like when you see a relic, a religious relic, and you know that it's the real stuff there, and it's the story of it. And Nicola in the article, she talks about, you know, when you go to the Hard Rock Cafe and you see a guitar, doesn't really mean much, but if it says Jimi Hendrix's guitar, then you get a feeling when you see it. So it's like, it's not just a representation of that stuff, it's the real stuff. It is safe. I've written a lot about the bacterial sublime as well, so it's to do with this kind of fear, but it is safe, and I can prove it's all safe. So yeah, I did actually handle live play at Porton Down as well, but that's on me. So and that was in a Category 3 lab.
Thank you. There's a question here.
Yes. Hi, I'm Dutch. My grandmother survived the Hunger Winter in Rotterdam.
Mm-hmm.
So I'm the third generation that we were talking about. So I was also thinking about how this story that you're talking about is my family history. And I have a bit of a hard time with this sculpture also when you were talking about the person in India. It resonated for me. But I was wondering how you as an artist navigate these sensitive post-traumas and these stories and these events, and then also mythologizing or like trying to navigate a story in that sense and work with these themes respectfully. Does that make sense?
I mean, from my perspective, I try to be honest about, like, this is what we know, this is how I've represented it, and these are the experiences. I do a lot of work in the Netherlands, so I know a lot of Dutch people. I've talked about, you know, things like the beans boiling on the side for hours, because that's the food that they have. And this idea, even now with fava beans being associated with poverty, even though they're trying to bring them back as a kind of the protein transition is the thing. And so in Bengaluru, I didn't really pull any punches. I talked straight away about the Bengal famine and different issues of famine, but asked them. I always asking other people to tell me things. It's like I want to learn, so I guess it's a vehicle for learning as well, for me. But just to be really honest about what I know and what I feel. And I think we all had a conversation that there was things about in our parents to do with like wartime or different experience, or like my mum was evacuated in World War II, and I grew up with all these stories about it, you know, like, if you don't eat your dinner there, I'm going to serve your dessert on the same plate, because we used to have to clean our plate and we'd have the dessert on the same plate when I was evacuated, she would say. And so if they had, well, at least they had dessert and stuff, they didn't have the famine. In fact, she told me she was never hungry, but she just didn't really like what they fed her. But yeah, it's quite traumatic. I mean, she ran away from being evacuated. She ran back to London because she hated it so much and didn't want to go back because they treated her like a slave, she said. And, you know, she had to do all the housework and everything and things. So I think we have our own traumas as well that we can kind of draw on in this. And it's just, I mean, I guess I'm just trying to be honest about.
Another questioner in the front row.
You talked a bit about like intergenerational trauma. I was like, I guess, how does that translate on, like, I guess when people are suffering from things like PTSD, how would that translate onto a separate generation that is like kind of feeding off of the PTSD from the generation before them? How would that be like displayed, and how could you actually cure that?
I mean, that might be.
Yeah, there's been, when I was looking into this and this epigenetics, there's been some studies with with sort of subsequent generations of Holocaust survivors, but also of generations. There was an interesting study of children of Vietnam veterans, and it was different to this because it's through the male line as well, so there's sort of different aspects to it. But they were finding that and I can't remember exactly what the experiments or the sort of the study consisted of, but they were looking at I think how they responded to, it's about cortisol levels and how they responded to stress, and found that there was a difference between the cohort of people whose parents had been or whose fathers were Vietnam War veterans than the cohort that hadn't. So they had a heightened response to stress. So yeah, there's not. I think there's not a great deal about it, particularly down generations, because you kind of got to wait until people go. Until you've got a cohort, which is why the Dutch winter famine study was so important, wasn't it? Because it was one of the first one that showed this sort of two generational impacts. But yeah, there was bits and bobs.
And we haven't come across anything that shows how you can kind of mitigate it or in your reconciliation work.
No, I mean in the work with, well, it's sort of more from the post-memory side. So Marianne Hirsch talks about the ways in which your responses to these cues, these visual cues that are given, can either lead to kind of heightened responses and trauma, or can lead to ways, or can be used in ways to mitigate it. And it sort of relates to your question too about the artwork and responses to it, because I think there's you create something, you create a piece of art, and it sparks conversations and responses, and people will respond in different ways, and you're not in control of that. This is an interpretation of something, and certainly when I do quite a lot of work in Bosnia particularly, and the conversations, I suppose it's easier to find a way in to having a conversation, and particularly having a conversation across different divides, if the prompt is something that's not related to the context in which you're talking about. So if the prompt is something else, so taking this to India and talking about the Bengal famine is different than if you took this to the Netherlands and talked about the Dutch famine. You have a different sort of set of responses, and people maybe feel closer to it. So I don't know, I think I sort of went off on a ramble with the topic there, but I was sort of thinking about these ways in which we respond, and some of the cultural cues that are layered into different artworks and into different prompts will be picked up by some people and not by others, because they'll be seen by some people and not by others. So different responses.
I think, given that we're nearly at 7:45 and there are drinks waiting for us in the next room, I just wondered if everyone would like to join me in thanking. And before we finish, I just wanted to read something that was published by Anna McNally in Studio International 2007-15. She was interviewing Anna, and she started by saying, not many artists would want to work with bacteria, smelly facial transplants, or anthrax, but Dumitriu is at the crossover between science and art, as happy in a microbiology lab as she is in a studio. I think that summed it up wonderfully. Thank you for sharing some of your brilliance with us tonight and explaining lots of things that many of us maybe haven't given a lot of thought to, and it's sort of illuminating in many levels. And we can continue the conversation over drinks, but firstly, let's just say thank you very much.